This bucket is actually how we have the eyes shipped to the Science Center. So this bucket is full of about 60 sheep eyeballs, all kind of floating around in a preservative liquid. Kind of smelly stuff, but your brain will actually desensitize to the smell over time. So if you're ever coming up on a dissection and you're worried about the smell, know that after a few minutes, your brain will start to ignore it. And so I'm actually completely desensitized to the smell already because I've had these eyes out on the table for a little while now. And so I really don't even notice the smell anymore. All right, let's switch our camera over here so you can take a look at some of the supplies that are on my tray. And as usual, I'll need to turn off my virtual background because it might cause us a couple problems as we start to move things around. Now, while I hit the lights to give us a little bit less of a glare, tell me, do you know any of these dissection tools that I have out on my tray? So these are both sheep eyeballs. Even though they look a little different, those are both sheep eyes. Tell me, do you know the names of any of these dissection tools? Go ahead and let me know in the chat. Great answers coming in. Some people are telling me that they see scissors. That's exactly right. The scissors are the most important tool we'll actually use in an eye dissection. It's really the only tool that you need, which is kind of cool about this dissection. I do have some other ones out here just in case we might need them as we move throughout the program. This is in fact called a scalpel. A couple people telling me that in the chat. Scalpels are extremely sharp. And actually, for the purposes of this dissection, I really don't need the scalpel. Scalpels are good for other types of dissections like fetal pigs or frogs, but we won't really be needing this today. So I'm actually going to put the cap on and just set it off to the side. So if I do need it, it's here, but we really shouldn't need the scalpel for any part of this eye dissection today. How about this one? I haven't really seen, oh yeah, a couple of people telling me they know the name of this one. This is called a probe. Probes are really important dissection tools. It'll help me to point at structures for you to be able to name or label. It also allows us to manipulate things that are a little bit smaller than my fingers. Since this isn't very sharp, we actually specifically call this one a blunt probe. All right, good job, everybody. You are pretty knowledgeable on your dissection tools. So before we start our dissection, let's talk a little bit about eyes. So I have an image here of a human eye, which we said is going to be very, very similar to this mammalian sheep eye that we'll be dissecting during the program. So name me some structures that you might know that you can see just from this picture of an eye. So we can't see all the inside parts of the eye just yet before we start our dissection or before we open anything up, but we can start by labeling some of these exterior or external structures of the eye. So if you know any of them, go ahead and tell them to me in the chat. Now, even things we might not conventionally think of as part of the eye are still part of the eye. And so really name anything in this photo that you know or that you might be familiar with. All right, so some guesses starting to come in on both Facebook and Zoom. Some people are telling me that they see the eyelid and the eyelashes. Those are important structures for our human eyes, and animals have eyelids and eyelashes as well. They're there to protect our eye. Since this is a very important organ that helps us to see, it's important that we keep our eyes safe. And so we've got the lid in order so we can close it to protect our eye, and the lashes to keep other things out as well. Some people even know some of these other structures. Some people are telling me that there is a tear duct somewhere over here in this region of the eye. You can even see some blood vessels in this white part of the eye. Lots of people pointing out things like the pupil, which is this dark spot in the center of the eye. So the dark spot we call the pupil, and you might know that the pupil actually changes size based on how light or dark it is in the room. this colored part of your eye, that is called the iris. So irises can be different colors depending on your genetic factors or your genetic traits. So if both of your parents have blue eyes, you are very, very likely to have blue eyes. Some people have brown eyes, some people have green eyes, and some people even have kind of a combination like we see in this eye, where we see some blue and even some maybe brownish or hazel tints towards the center. So this diagram is labeled. So we've got some of those labels on here. We said the dark spot in the center is the pupil. We then have the iris, which is the colored part of your eye. And then the white part of your eye is called the sclera. And the sclera is a super, super tough tissue. So let's take a look at our sheep or mammalian eye. And so the sclera is the white part that kind of surrounds the entire eyeball. We said it's very, very tough. It's there to help keep our eyes shape. And we're actually going to have to remove a little bit more of this tissue that's surrounding our eye in order to get a good look at some of our eye structures. So now you'll see just how important these scissors are during this dissection. So our scissors, or our snips here, are going to allow us to kind of trim off some of that extra kind of connective or muscular tissue that surrounds the eye, right? So we know that there are muscles around the mammalian eye that allow us to change the direction that our eye is facing. So even without turning our head, we can turn our eyeballs even within our head. Now, this part of the dissection really doesn't need to be done very delicately. That's because the sclera is so incredibly tough. It's really, really hard. We said it's there to give our eye structure. And so it's actually hard to accidentally puncture the sclera unless you're really, really trying to do so. So you'll notice I'm not being very careful, just trying to trim off as much of that extra tissue from around the eye that we don't need. So you can see we've got some of that eyelid here left on from when this eye was removed from the whole specimen or the whole animal. And we're just going to trim all of that off. All right, so now we have a pretty good look here at our mammalian or sheep eye. To talk about the structures in which directions we're pointing, it's really important that we understand body directions or orientation. So this obviously would be the front of the sheep's body, or the front of the eye. On the back, we have the optic nerve, which is this piece of tissue right here that kind of pokes off the back. In science, though, we really don't use the terms front and back. We use something a little bit more scientific. So we'll take a look next at this picture. Oops, we'll go to this one. Where are our directions? There they are. All right, so these are our body directions. And you can see that in science, when we're talking about the front of an organism, we usually say anterior. So anterior means front, whereas posterior means back. So if throughout this lesson you hear the phrase anterior and posterior, remember that anterior means front and posterior means back. Now we have names for all the different directions of the human body. So there's superior, which is towards the top or your head. We have inferior, which is down by your feet. We've got lateral versus contralateral. But really the most important ones we'll talk about today are this anterior versus posterior structures of the eye. So just keep those terms in mind as we move through this dissection. Had a really good question already come in through the chat. So if someone asked, is this eye real? And it is real. So this is a real sheep eye that we have to dissect here. So it's not made of plastic. And you'll see as we cut into it, you'll be able to tell just how real this eye is. So we'll scoop some of that excess tissue that we're not using away, and we'll be making a cut through our eye that separates it into anterior and posterior sections. Another great question we should address as we move through this program is that we did not kill this sheep. Someone did ask, did we kill the sheep? We absolutely did not do that. Like we said, we ordered these eyes from Carolina Biological Supply, and so we did not kill the sheep. We'll keep talking about this as we cut our eye. So from anterior to posterior, we really want to split it in half. Now, these sheep eyes came from a slaughterhouse, meaning that these sheep were killed for food. So people ate these sheep. They didn't just kill them just to take their eyes for the sake of dissections. That wouldn't be very ethical. You can find out a lot more about where dissection specimens come from on the various supply websites. They always post information about where they come from. Keep in mind that someone did eat this sheep for food, so it really served a purpose in life before we just got its eyes kind of supplied to us for the use of this dissection. So don't worry. All right, so I made a cut all the way around our eye. You can kind of see it there, especially if I hold it a little closer. So I made a cut so that we can separate the eye from anterior, the front of the eye where our cornea is, to posterior, where our optic nerve carries all the information from our eye to the brain. So anterior, posterior. All right, let's go ahead and we'll just separate the two sections here. And you can see that there's all sorts of stuff here inside the eyeball. So there's a couple more structures holding it together. We'll go ahead and just get a final separation there. That way we've got our, remember, anterior and posterior parts of the eye. Now, the first thing we notice is that there's all sorts of jelly stuff here inside the eye. So let's take a closer look at that. All right, so this jelly stuff is called the vitreous, and it really is like a liquid, so it kind of holds its shape, and that really fills the back side or posterior side of the eye. Now, if we remove some of that vitreous humor, that gel-like substance from this section of the eye, we'll get a close look at one of the most important structures in the sheep or mammalian eye. We want to leave that structure in place, so we might just need to do a little bit of a dump here. All right, so we've dumped out most of that vitreous humor from the anterior section of our eye. And does anyone know what this little structure is called? Does anyone know? Anyone familiar? What is this gumball-like structure called? So this is actually, it does look like a grape. That's actually called the lens. Right now, this eye has been preserved in a liquid, which allows it to stay fresh enough for us to dissect even months or really even years after it was killed and preserved. Now in life, this lens is really, really clear. It's really clear, and so that way we can see through it. So we'll go ahead and we'll take the lens out of our eye, leaving that anterior section of our eye just right there on the table. And so this gumball-like structure is the lens. And the lens is actually the part of our eye that changes shape in order to allow us to see things that are either really, really close or really, really far away from our eye. Take a look at a diagram of exactly how that works. So we can see that this black dot here in these diagrams, that is the lens. So that lens right there can change shape. So between our two diagrams here, if we are focusing on something that is near to our face, something that's up close, the lens is pretty wide, or what we would call globular. If we want to shift our vision to something that is farther away, the lens will flatten. And in order to do this, our eye has these muscles that surround the lens. So in life, the lens is surrounded by these ciliary muscles. They almost look like little hairs, which is why they're named ciliary muscles. So if you're familiar with your cell structures, these little hair-like projections that come off of cells are called cilia. And so that's why these muscles got their name, because they look like those ciliary hairs. And so those muscles are able to pull really tightly on the lens in order to stretch it so that our eye is able to focus. So you can actually see those ciliary muscles or those ciliary bodies here in the anterior or front section of our sheep eye. So if we take a close look, hold my hand still, so hopefully our camera will focus here. There we go. So right here, we can see those hair-like muscles. So we removed the lens, but before we removed the lens, these ciliary bodies or ciliary muscles were really connected to that lens. You can see them even better if I tilt this to the side and kind of catch the glare from the light. So these ciliary muscles allow our lens to change shape so that our eye is able to focus. Now, this part of the eye right here, this opening, does anyone know what this opening is called? Looking at it from the back is kind of interesting because we're pretty used to seeing this structure from the front. Does anyone know what that opening is called? Think about it. What would you call the opening in your eye if you were looking at it from the front? Remember that the structures are the same. We're just looking at them from a different orientation than we might normally. So this is actually the pupil. So that is the pupil of a sheep eye. Now, this is one of the big differences between a sheep and a human eye is the shape of this pupil. Now, on a human, maybe if you have a parent or a friend or a sibling sitting next to you, take note of the shape of the human pupil. This one is not the same. So human pupils are round, whereas sheep pupils are much more rectangular, almost like an oval. So they're very, very long across the eye and not very tall, whereas human eyes are round. And this is actually a distinct feature of most prey animals. So sheep are prey. They don't do a lot of hunting for food, so they are not predators. More commonly, they're eaten by other animals, and so we would call them prey. Prey animals have really long pupils so that they can almost see like a panorama. They can see really far to either side, but they can't see very high up or down. That allows them to be able to see predators coming from all the way around the side. So they have almost a panoramic field of view long ways, whereas predators don't really need that because predators are the ones doing the hunting. And so these prey animals have adapted this structure over time. All right, let's switch over here and let's talk about the posterior or the back side of the eye. Now I'm going to very carefully this time try to dump out that vitreous or gel-like substance, that vitreous humor. There we go. And I've tried to leave this structure almost completely intact on the back of our eye. Now, this kind of skin-colored flap of skin, you'll notice that if I touch it, it almost moves around, or it's really easy to move around. That is the retina. So if you've ever heard of someone tearing their retina, you can see just how easily that might happen because this tissue is extremely thin and is extremely delicate. So this is the retina, and it's stretched all the way over the posterior side of the eyeball. That's actually a flap of skin that's covered in cells, millions and millions of tiny cells, which allow us to see. So this is a sheet of tissue covered in cells that we call rods and cones. So if you've heard of rods and cones, I'll show you another picture of what those look like. So if you recognize this image, it was my virtual background right when we started the program. And so if you could take an up-close image with an electron microscope of this retina, you'd be able to see these different cells, which are called rods and cones. These are actually the cells that give us vision. So when light travels through your eye, first through the cornea, then through the lens, then through the vitreous humor, that gel-like substance, that light then strikes the retina on the back of the eyeball, which sends information to these two different types of cells. Rods are what give us differentiation between light and dark. So rods tell us how much light is coming in, so they monitor brightness, whereas cones tell us what color the light is coming in as. So we've talked about wavelengths of light a few times here on Echo Live. We've talked about frequencies of visible light. And so these cone cells, which you can see really clearly in the electron microscope image, these cone cells pick up different frequencies of light And that's what allows us to be able to see color. All right, so we've got rods and cones, and those make up the retina, which is this flap of skin that covers the entire posterior side of the eye. Now we'll go ahead and we'll just gently kind of push the whole thing off to the side because there's something really interesting about the retina. Now, if we kind of keep pushing it around, you'll notice that the retina comes to a point. So it's attached to the eye at only one place. So right here, that entire flap of skin is connected to the posterior side of the eye. Now that is actually what we call the blind spot. So if you've ever heard of a blind spot, it is real. So all mammals have a blind spot where the retina attaches to the back of the eye, and where all of this retina, where all of these sensory cells exit the eye, comes right out to the optic nerve, which you can't see very well here on the camera. Let me see if I can get our camera to focus a little bit better. All right, so you can see that from our retina, all of that flap of skin feeds into the eye and comes out the other side as the optic nerve, which carries that information to the brain. So on that one tiny spot on the eye, we actually don't detect any light or any color. That is really our blind spot. We don't get any information from that spot where it all feeds to the optic nerve to be carried to the brain. All right. Now, is there anything else kind of interesting that you notice about the back or the posterior side of a sheep eye? Tell me in the chat, is there something interesting that you notice? Something that you maybe think is kind of strange or cool looking? So I'm talking specifically about this structure or this tissue right here. So this tissue, which you can see almost looks like a seashell. Yeah, someone in Zoom tells me that it almost looks like a rainbow. And you're exactly right. Now, this is a structure that also makes sheep eyes unique from human eyes. This structure is called the tapetum lucidum, and it's very, very reflective like that, which allows sheep to have really good night vision. So it takes the light that's sent into the eyeball, it reflects it around, and then we can pick up even more light than you'd be able to if the inside of your eye was dark. So only animals with night vision have this tapetum lucidum, this really rainbow kind of reflective layer of the eye that's located behind the retina. So cats have this, cows have this, sheep have it. Really any animal where you look at their eyes and it almost glows back at you at night, they have this layer called the tapetum lucidum. Humans don't have particularly great night vision, and so we don't have this layer of our eye. This is, again, something that is pretty unique to certain types of animals over others. Humans just don't really have a need for great night vision, and so it's not something that humans have developed over time. All right, now I'd like to take some questions. So if you have any questions about our eyes, I have one that I dissected earlier, just here as an example. This one has more of its retina intact. If you have any questions about these eyes, go ahead and tell me in the chat. I see a couple really great ones already. Someone asked, how do people tear a retina? Or how do we know if we tore a retina? Those are both great questions. People can tear a retina in many ways. One of the most common ways is from trauma. And so if you bang your head on something really hard or something were to enter your eye, you saw that that flap of skin is extremely delicate and it's really not being held on by much. So it's really just held in place by that vitreous humor, this jelly-like substance that you see is now dumped out on the tray. And so if you have a trauma to your head, your retina can either dislocate or it could rip inside your eye. A sign that you might have a torn or a damaged retina is actually if you're seeing bright flashes of light in your eyes for no reason, even if you know they're not coming from the environment around you. If you're seeing lots of bright flashes of light, that means that this retina, which has all of those rod cells on it, could either be folded up or torn, and so it's sending incorrect information from your eyes into your brain. So if you are seeing bright flashes a lot, more commonly than maybe you know you used to, that could be a sign that you need to have your eyes checked because maybe you have a damaged retina. Jake asked, where is the iris? That's a great question. So the iris right here on this side of our eye is this layer around the pupil. So the pupil is the hole and the iris is the layer around it. So the iris in humans is what give us different colored eyes. And we know that that iris can also, it's full of muscles and can actually change its shape in order to allow more or less light into our eyes. So the iris in sheep are all pretty much the same black or really, really dark brown color. But in humans, genetics give us different colored irises. Good question. Let's see. Someone asked a question about if you exercise your eye muscles, can you avoid ever needing glasses? No. So there are a lot of myths associated with the eyes and how our vision can change over time. And most of it is really just based on age or on use of your eyes. It's a little dark in here because I turned off my studio lights. But one of the biggest myths associated with eyesight is that if you spend too much time squinting with your eyelids, that it can actually damage your eyesight. And that's actually not true at all. What it means if you're squinting a lot is probably that your vision is already going bad and that your body is trying to compensate by focusing your lenses yourself. But we really can't do that. In order to focus our lenses, we really rely on those ciliary muscles. The muscles really have to pull and release on your lens in order to change the shape. So squinting doesn't really do anything to help you with your vision, and it actually can't damage your vision at all. But what the danger of squinting too much is that it could give you headaches because you're using a lot of muscles around your eyes, which might make you think your vision is going bad when it's really not. But that's a great question. A couple of people saying that they know people or have personally had a torn retina. I know that my mom had a scratch on her cornea, which is the front part of your eye. So if we turn over our anterior or front section of our eye here, this is the cornea. And it doesn't look great because the preservative makes it really, really cloudy here in this sheep eyeball. But if you were to scratch your cornea, right, the light still needs to enter your eye. Thankfully, your eyes are one of the fastest healing tissues on the entire body. I actually think it might be the second fastest healing as opposed to your tongue. Just because of how much blood flow comes to your eyes, they heal really, really fast. And so damage to eyes, thankfully, as long as it's not too severe, will heal pretty quickly. But having a scratch on this part of your eye could impair your vision pretty badly. There are lots of kind of ailments or diseases that come with eyes. If you know someone who has a lot of floaters in their eyes, so things that kind of look like little hairs floating around, that happens with age. That's a natural process. All that is your vitreous humor, this jelly substance, starting to age, which will cause the proteins inside it to clump up. Now, this vitreous humor is cloudy already, and that's because of the preservative that we put in these sheep eyes. But in life, this vitreous humor is clear, totally clear, crystal clear. But if you're seeing floaters in your eyes, that's normal. You get more of them as you age, but that's just because that vitreous humor is starting to break down over time, and you can actually start to see the proteins floating through your eyeball. Some people ask what causes blindness, and that could be from a variety of things, right? If you completely dislocated your retina or it was detached from your optic nerve, that part where the blind spot leads into the optic nerve on the posterior side of your eye, if it was completely detached, there are no sensory cells in order to send information to your brain. And so that could result in blindness. You could also have someone who has severed their optic nerve completely. So if the optic nerve is damaged on the posterior side of your eye, If that were to become detached or severed or damaged in any way, that could cause blindness. So there are all sorts of structures of the eye, and each of them has different kinds of problems associated with them. So there's not one cause of blindness. Some blindness is caused in the brain itself, and it actually has nothing to do with the eyes. So that's a great question, but every structure inside your eye serves a purpose. So if one of them were to be damaged, it's likely that that could result in either partial or full blindness. Lots of great questions coming in. David asked that he thinks that I know so much about everything, and that's not true. I don't know a lot about everything, but I do have a good background in lots of things science related. So like I said, I studied biology when I went to Wayne State, which is right across the street from the Science Center. So I studied biology, and when you study biology, you also have to take courses in chemistry and in physics and in psychology. And so you get a good span of knowledge when it comes to science, especially if you study like something like biology. So I'm very, very lucky, and I find science all to be pretty interesting. So I was glad that I didn't have to choose just one specific topic to study. A couple people asking about other resources. So if you're looking for more resources, unfortunately, getting sheep eyes to dissect at home is kind of tricky. You do have to order them from a scientific supply company. So it's not super easy to come by. You can if you're interested in doing so, and they often will come with dissection guides. If you are looking for at-home science experiments that you can try, maybe not necessarily related to dissections, of course, you can visit our at-home science website. We're updating that just about weekly. So the last experiments that were uploaded are from Earth Day and are Earth and space related, but we'll have even more activities coming out this week about some of the experiments that you'll see coming up on Echo Live throughout the week. I know we're a bit over our time today, and that's just because I love dissection so much that I have talked and talked and talked your ear off. If you have more questions, send them in the chat. I'd be more than happy to answer them throughout the day if you do have other questions that we didn't get through here on Echo Live. We'd love to have more dissection labs, so tell us what you thought. Did you enjoy or not enjoy the dissection lab? I'm hoping people enjoyed them so we can keep doing more. I think they're very fun and interesting, and we can always dissect something else next time. All right, so thank you everyone so much for joining us. We hope we'll see you back here tomorrow at 2:30 p.m. for more Echo Live shows. We'll be talking about something unrelated to dissection, so a little bit of a change up. We hope we'll see you back here. Big thank you as always to our sponsors who allow us to bring these programs to you at home for free every single weekday. If you're enjoying them, make sure that you are liking, following the Michigan Science Center on Facebook, and feel free to share these videos with your friends at home. Thank you everyone so much for joining, and we hope we'll see you back here tomorrow.